Hey, general biology class. After we concluded class yesterday where we worked with protein structure, I realized that the terminology that we used during class was not consistent with that in your textbook. And so I am going to go over the same material briefly here and make sure that the terminology we use is consistent with your textbook. As always, if you have any questions about this, you can feel free to email me, and I'll provide my email address at the end of this recording. You can also stop by my office during my office hours or at any time. So, as you remember, yesterday we used this sequence to form a polypeptide, and each of you had a card, and we formed a primary structure that looked like this. So then you lined up in the classroom, and then we talked about forming the different interactions. Slides. However, as I said, we have some inconsistencies with the textbook. So for the next few slides, I have corrected the information. So the primary structure stayed the same. You formed a peptide bond by placing your hand on the shoulder of the person in front of you in the molecule. That remains the same. And this was a diagram that was supposed to show the order of how you stood in line. Somehow in the importing process it became distorted, so we'll move on. However, then we talked about the secondary structure several times, and that is where the inconsistency lies. Secondary structures were not represented in our example yesterday. Secondary structures involve hydrogen bonding within the primary structure of the protein, and they take two forms. You either have an alpha helix, which is represented here, or you have a beta pleated sheet, which is represented here. Again, these are not caused by covalent bonding at all, but they do occur as polypeptides, the R side chains, the ones that are different between each amino acid, interact to have hydrogen bonds. What we did do was a tertiary structure, and these result from interactions between amino acids and there are groups within that side chain. And so you can see some examples right here of two different interactions. We actually have a hydrogen bond here within the same polypeptide. We have hydrophobic and van der Waals interactions occurring, possibly, but we also have disulfide bridges, which we're about to show an example of. Again, this is in the tertiary structure. It is providing a shape to a single polypeptide chain. So our slide should have looked like this yesterday when we talked about in the cysteine amino acids being attracted to one another, and the sulfhydryl group, which was represented as SH, a sulfur and a hydrogen, can form a bond, and that is part of forming the tertiary structure. And so actually this is what it looked like when we accomplished that during our class meeting. We also had weaker ionic interactions where we had oppositely charged regions, and that again was part of the tertiary structure. And so here's where we had the two groups of students, some positive and some negative, interact to fold the polypeptide into another bend in that tertiary structure. And then finally, we looked at the hydrophobic interactions where we clumped together the nonpolar side chains, again, in the tertiary structure, where you can see that on the other end of the disulfide bond, we hid those nonpolar amino acids within the clump of the tertiary structure. Now, just like any example, this one is not perfect. So, things that were not represented in the actual construction of a complete protein in our example were, as we've already said, not the secondary structure. You can see the above slides that I've already mentioned and gone over about how we did not represent the secondary structure. We also did not represent completely the chaperonins, the proteins that assist in the folding of other polypeptide chains. Now, Bradley and Megan, who didn't have cards, tried to assist at least one of the polypeptide chains in their folding. However, chaperonins are very large and the entire polypeptide fits inside almost like fitting into a barrel. And they clearly did not play that role completely, but they did assist each, or one of the teams anyway, in folding properly. One other thing to think about is that protein folding occurs in 3 dimensions, and we were only able to fold in 2 dimensions. Had we had strings from the ceiling or items like that, perhaps we would have been able to simulate this in 3 dimensions, but as you know, we didn't have those things, and so we only were able to simulate in 2 dimensions. And then finally, we did not represent in our example the quaternary structure, and that would have been where we could have linked the 2 polypeptide chains that we built into one cohesive whole protein. We mentioned how hemoglobin has 4 subunits, and those 4 together form the entire hemoglobin molecule. That would be an example of the quaternary structure. As I mentioned, if you have any questions about this or anything else, you can come by my office. You can send me an email. My email is sjohnson@swu.edu. I'll be happy to answer your questions. I will be happy to go over this again if needed, or to talk about anything else that you'd like to discuss. I hope you have a great weekend, and I will see you next week.